Hello, Bio. Welcome to our video lecture for chapter 23, Broad Patterns of Evolution. So the picture I picked for this chapter might just look like a bunch of big rocks, but these are actually the oldest fossils on planet Earth. They're called stromatolites. They're fossilized mats of bacteria. We'll talk about them in just a little bit on a later slide. This chapter is a pretty simple chapter. There's some important concepts to discuss, but compared to some of the other more detailed chapters we've had, this one hopefully you'll find pretty simple. So we've already defined the word macroevolution. This is back in chapter 19 and 20, sort of a broader Darwinian view. Questions that this might answer, things like where do terrestrial vertebrates come from, the impact of mass extinctions on evolution on planet Earth, where flight and birds came from. Remember, microevolution was looking at allele frequencies. That was how do you mine bird. Macroevolution is obviously on a much larger scale. So the fossil record, we've discussed before how, particularly with sedimentary rocks, you get the strata, the different layers. And sedimentary rocks are the most common rocks where you find fossils. And obviously, if you want to study, evolution in the past, studying the fossil record is a good place to start. Some disadvantages of the fossil record. So not everything fossilizes. Things that existed for a long time, things that had hard parts like shells or bones, things that were abundant and widespread are more likely to leave fossils than things that didn't have bones or shells or were around for a much shorter period of time than other things. So the fossil record is great, but just recognize it's going to be incomplete because not everything fossilizes. I mentioned this just a minute ago. The oldest known fossils are called stromatolites. They're found in sort of shallow waters. They date back to about 3.5 billion years ago. So, the current thinking for a date of the origin of life is about 3.5 billion years ago. Could there have been things living 3.6 billion years ago? Sure. Maybe they didn't fossilize or we just haven't found them yet. Remember, Earth is about 4.6 billion years old. So the first billion or so years of Earth's history, there was no life. At least nothing that left fossils that we have found. This is a cross-section of a stromatolite. Basically, it's sort of like the idea of coral reefs built upon layers, but in this case, they're bacteria, not coral. So relative dating. So, typically speaking, we've discussed this before. The deeper down a fossil is, the older it is compared to fossils on more surface strata. So if I find a fossil on a layer here and find one on a strata beneath it, the one beneath it's probably older. Index fossils are ones that are widespread and you can use to date how old a layer is. So say you're at a strata and you find a fossilized trilobite and you can date it to 70 million years ago. You can assume that layer is about 70 million years old. Sometimes relative dating, that doesn't give you an exact date, but it can tell you like a younger than, older than type thing. Geologic time scale, this isn't something you need to know. It's just kind of interesting to talk about. Let me move my face real quick. So the main epochs in Earth's history. So on this graph or this chart on the right-hand side, this is millions of years ago. So right now we're in the Holocene. The four major time periods are the Cenozoic, the Mesozoic, the Paleozoic, and the Precambrian. You subdivide these into other epochs or other periods, I should say. The boundaries of these periods usually represent mass extinctions or sort of massive changes in Earth. The Cretaceous period, going from Mesozoic to Cenozoic, the Cretaceous tertiary boundary is the boundary. It's called the KT boundary, where the dinosaurs went extinct. We'll discuss that later. The Permian extinctions, going from Paleozoic to Mesozoic, Permian extinctions were actually more things went extinct then than when the dinosaurs went extinct. We'll come back to that though later. The AP exam could give you this chart and ask you questions about it, but the names you don't need to have memorized. This is just another picture of the geologic time scale when humans appeared. If you want to pause this and look at this, feel free, but these aren't things you need to have memorized. So absolute dating in comparison to relative dating. This is using radioactive isotopes, particularly carbon-14, to actually date when a fossil existed. So really quick, so we discussed this back in like chapter 2 or 3. So the main isotope of carbon is carbon-12. Carbon-14 is a radioactive version of carbon-12. Radioactive elements have what's called a half-life. It's basically the time that it takes for half, whether it's 8 milligrams or 8 grams, the time it takes for half of it to decay back to the normal isotope, or the more common isotope, I should say. And for carbon-14, it takes about 5,600 years for half the sample to decompose back to carbon-12. Most of the carbon in your body is carbon-12. You do have some carbon-14 though. Carbon-14 is made on Earth in the upper atmosphere. Basically, when carbon dioxide, CO2, which you breathe in, UV radiation from the sun can turn carbon-12 into carbon-14. So some of the CO2 that you're breathing in right now might have come from the atmosphere, and some of it might be carbon-14. So as long as you're breathing, as long as you're alive, the ratio of carbon-14 to carbon-12 is the same as it is in the atmosphere. Now, once you die, you stop breathing, right? So your carbon-14 starts to decay back to carbon-12. So basically what we do is we look at the ratio of 14 to 12 in a fossil compared to the atmosphere, and basically it tells you when the fossil died or when it stopped breathing, which is basically when it existed, right? This graph just shows the way half-lives work. Every half-life for carbon-14 is about 5,600 years. So, if it's been between 11 and 12,000 years, you know that 2 half-lives of carbon have passed. So if you had 8 grams and now you have 2 grams, you cut it in half twice. This is actually a good question. Say you had 8, let's use milligrams. Say you had 8 milligrams and you're down to 1 milligram. How many half-lives is that? From 8 to 1. Each half-life, you're cutting it in half. So how many times you cut 8 in half to get to 1? So 8 cut half once is 4, then 2, then 1. All right, so you cut it in half 3 times to get to 1. That means 3 half-lives have passed, which would be around 15,000 years or so. It's the 5,600 times 3. C14 dating is good up to about 75,000 years old. Then it's just too small to detect. There are other isotopes you can use to date things that are older. You can date when volcanic rocks hardened using other radioactive isotopes besides carbon. But if something died 60 million years ago, the carbon-14 dating would not be able to date it. You'd have to date what layer the fossil was in. So we're just going to briefly discuss this. So development in macroevolution. So, the idea that every developmental change between species is actually a different gene, that's very cumbersome. That would require a bunch of different genes. Remember, you and a chimp are about 98, 99% the exact same DNA. So a lot of times you can have the exact same genes, but how they're expressed and how they're expressed as the organism develops can change, which can lead to huge differences in what the animal looks like. So 2 examples, allometric growth and heterochrony. So allometric growth, I know this diagram looks really, really creepy. This is basically differences in relative growth rates for various parts of the body. So look at like a 2-month-old fetus, the head's like half the body, right? Look at a 25-year-old adult, the head is like, I don't know, an 1/8 or a 1/7 of the body. So basically you can have large changes in appearance or development just by changing the growth rates of different parts of the body. Heterochrony, so look at this picture. Look at a chimp fetus's skull and a human fetus's skull. They look very, very similar. Look at the adult versions, they look very different. So basically, you don't have to have that many different genes. You just change when and how the genes are expressed, and you can have huge developmental differences. These words, allometric growth and heterochrony, you should know that they're talking about changes in development by changing relative growth rates, like from a fetus or an embryo to an adult. This just shows it. So, a bat's hand and finger bones are similar to yours, but obviously they're expressed way more compared to the body than they are in a human. Homeotic genes, we mentioned this, back in chapter, I want to say 15, one of the teens. So homeotic genes, remember, these were the genes. Remember the picture of the fruit fly with the legs growing out of its eyes instead of antenna? These are genes, and a Hox gene is an example. These are genes that code basically for body formation, for pattern formation, for like where you should put the legs. So, an insect has 6 legs. A spider has 8 legs. Do you need a bunch of different genes for that? Not necessarily. You could just have changes in homeotic genes, which tell you how many legs there should be or where you put the legs. An example, these are 2 different kinds of arthropods, fruit fly and whatever, this looks like some kind of crustacean. You know, you don't really have to change the gene for legs. This is showing the Hox gene number 7, whatever that is. And basically changes in that gene give you the different leg patterns. So again, I don't need a gene for every different type of leg. I can have the same gene and just change the gene for pattern formation, homeotic genes like the Hox gene. So I have a feeling we probably discussed sticklebacks already. This is an activity that we do usually when we do Hardy-Weinberg in class, but this is a good case study. So sticklebacks, and this case study is taking place in Alaska. Sticklebacks are little fish. They look like this. Sticklebacks can live in the ocean or they can live in freshwater. They can live in freshwater ponds or they can live in marine environments. You know, marine means the ocean. And what's interesting is sticklebacks that have these little spines, they also can have plated armor on the side. Having the spine is beneficial if you live in the ocean. In the ocean, there's lots of very large predators. And not many places to hide. All right, so the spines basically, like if a predator comes and swallows the stickleback fish, they're only about like a couple inches long. The spines will stick in the predator's mouth and they'll spit it out. In a freshwater environment, the spine is actually bad because in a freshwater environment, there aren't large predators like in the ocean. And the lakes in Alaska, the main predator of sticklebacks are, I believe it's the larva of dragonflies, and they're not that much bigger than the sticklebacks, but the larva can grab onto the stickle and grab it and eat it. So having the stickle is bad if you're a freshwater stickleback. So the question is, like, what's the genetic difference? And you would go into this thinking, well, there's a gene for having the stickle, and there's a gene for not having the stickle, right? That's actually incorrect. Both versions, the marine version and the saltwater version, the gene's called the PIDX1 gene. The gene's exactly the same. So the ones that don't have the stickle still have the gene for the stickle. The difference is, and hopefully we've already discussed this in class, is a gene ahead of the PIDX1 gene that acts as a switch and turns the gene on or off. It's like a regulatory switch. This picture shows the blue stains. Basically, they're looking for messenger RNA from the PIDX1 gene. If you have messenger RNA, you know the gene's turned on. So the marine ones that have the stickleback, it's turned on to the abdomen where the stickles form. In the freshwater version, the mouth part still have it, but the abdomen part doesn't have it. So again, the gene is the same. The mutation was in the switch in a regulatory gene that turned the gene on or off. So again, you don't have to, you know, if you lose your stickles, you don't have to lose the gene for stickles. You could just have a mutation in the regulatory switch that turns the gene off. So again, minor changes can have huge differences in phenotype. So quick word, you discussed Pangaea back in middle school. So we need to briefly just discuss Pangaea because it leads to some stuff about speciation. So 250 million years ago or so, when most of Earth's land masses were combined in a supercontinent called Pangaea, it's about 250 million years ago when Pangaea starts to break up. Today we have very deep, very dark oceans. Back in the day, the oceans were shallower. The climate was colder and drier, and there was more like shallow water habitats versus today. And you know, life likes shallow waters because you get plenty of sunlight. The waters tend to be warmer because it doesn't get as cold. When Pangaea breaks up, we're not going to get into the layers of the Earth. This just shows the major plates. No, I'm not going to worry about this. Pangaea breaks up about 250 million years ago, excuse me. Just FYI, the date for the asteroid killing the dinosaurs is about 65 million years ago. So Pangaea was way before the dinosaurs. So when Pangaea broke up, it led to a lot of allopatric speciation. If you remember back from chapter 22, allopatric speciation is when two populations are separated by a physical barrier. The example that I want to give is a really interesting example of this case study. about these two frogs. I'm not even going to try and pronounce those because I know that I'll mispronounce it. But the first frog is native to Madagascar, which is right here off the coast of Africa, right? This frog, the purple one, is native to India and Southeast Asia, which is right here. They're not near each other now. But if you look back at after Pangaea was breaking up, Madagascar and India were much, much, much closer together, such that the frogs could go back and forth. Now, what's so interesting here is on this slide, they're showing you two different ways of dating when these islands separated, or when India back in the day was not attached to Asia. So this method is using molecular clocks. If you think back to chapter 20, I believe, we discussed molecular clocks. We use it to date when viruses switch hosts. We discussed HIV and COVID-19. So if you count the mutations between these two frogs and use the average mutation rate of DNA, the common ancestor would be about 88 million years ago, looking at this cladogram. Okay, so that's one way using molecular genetics to date when species diverged. Using Pangaea and plate tectonics, this is 88 million years ago when the two were practically touching, and then they started separating when India went towards Asia. So geology tells us they separated about 88 million years ago. So it's two completely different methods of dating something that are giving you like the exact same number. And this is a good sort of theme. When two different ways of thinking give you the same answer, that answer is probably pretty reliable. Other examples, there's similar fossils in Brazil and the west coast of Africa. We discussed freshwater crocodile type things that weren't swimming in the ocean. Those two land masses would have been next to one another in Pangaea. Unique and diverse forms of life in Australia. So Australia has been isolated from the rest of the other land masses for a very long time. Can we go back and look at when Australia separated? So Australia, let's see, it's pretty separated. Gosh, going to 65 million years ago, it's pretty isolated. It's next to Antarctica, but we're not going to worry about Antarctica. It's just too cold. So it's been separated for a very, very long time. So allopatric speciation would dictate that those things have evolved in way different ways than other life forms. And because they're isolated, the gene pool is separate. Mass extinctions, let's end on a high note. So the fossil record indicates that most things that lived on Earth have gone extinct. There have been 5 major mass extinctions in which 50% or more of life on Earth disappeared. And this isn't a graph to memorize, but this just kind of goes through them. The red is the extinction rate, and the blue are the number of families. And why it shows you both, we'll discuss in a minute. So this one, where you had the most extinction, was the permanent extinctions between the Paleozoic and Mesozoic eras. What caused that, we actually don't know. It could have just been the climate change. The Mesozoic Cenozoic barrier, this is the Cretaceous period right here. This is the one where the dinosaurs went extinct. That one, which is probably caused by the asteroid, we'll discuss that in just a minute. So what's of note is what happens after a massive extinction. After it happens, what happens to the number of families? It starts to go up. So the theme here is that when lots of things go extinct, the species that survive suddenly have way more resources, all right, because there's not as many competitors. So they start to take over in a term called adaptive radiation, where they can adapt to new environments they couldn't be in before, and they just kind of take over the planet. An example would be how mammals took over after the dinosaurs went extinct. Around the time of the dinosaurs, they were mammals, but they were like little mouse gopher size things that were very small. They could hide easily. The dinosaurs obviously many of them were very fierce predators. When there weren't primates with the dinosaurs. When they go extinct, those little mice and gopher size things, their predators are largely gone. So they suddenly radiate and adapt out to new environments, and the age of the mammals begins when the age of the dinosaurs ends. So we could probably thank our existence to that asteroid. The evidence for it, and we'll probably talk about this in class. There's a video that I usually show. So the whole asteroid impact theory is only a couple decades old. I want to say it's in the 80s when it first became kind of a thing. So it's a relatively new theory. So there's a layer called the KT boundary. It's a layer in the soil. It's one of these strata that's found across the entire planet. And in that layer, there's a thin layer of iridium, which is an element that is mainly found in space objects like asteroids. So the idea here is that an asteroid hits the Earth about 65 million years ago. Think a gazillion nuclear bombs going off at once. The asteroid just gets pulverized. There's a crater in Chicxulub, Mexico. It's part on the Yucatan, part of it's sort of underwater. The video that I'll show you in class goes through this more. That's where the impact was. And when the asteroid vaporizes in the massive explosion, of course, if you were like at the impact site, you died. But all that dust that gets kicked off from the asteroid, which contains some iridium, basically for a period of months or years just blocks the sun because it's too dusty. And imagine, say you have a year of 10% sunlight compared to what you normally have. A lot of things are going to go extinct. Your big meat-eating dinosaurs, the food chain just collapses. Without a lot of sunlight, things start dying, and the animals that need the most resources, the most calories, are going to be the first to die. So big meat-eating dinosaurs, their food source goes away, so they die. That being said, some things could survive that. If you live in the ocean, you could survive that. If you're some kind of burrowing mouse, you could survive a year of 10% sunlight. And once all the dust settled, hence you get the layer of iridium around the entire planet, life starts to come back, and the things that survive the impact take over the world, hence the age of the mammals. All right, I mentioned this is our last slide. So mass extinctions paved the way for adaptive radiation, which is just the idea that you have a common ancestor, and that ancestor just adapts to way different environments and takes over. And the best example is the idea of the Cretaceous extinctions, where the dinosaurs going extinct, how mammals just radiated across the planet and adapted in different ways. And one of those lines eventually evolved into Homo sapiens. Okay, so that's chapter 23. Like I said, it's pretty short, pretty simple, and we'll see you guys next time.